Hi there, everyone. My name's Tom Rollins. I'm the marketing manager here at FORMULATRIX. I'd like to start out by thanking everyone very much for taking the opportunity, for taking the time out of your busy schedules to be here today. I'm extremely excited for this application-focused web event. So before we begin, I'd like to provide an overview of our agenda here. First, Paul Ventura, our Director of Sales at FORMULATRIX, will briefly introduce solutions for positive displacement microfluidic reagent dispensing for high-throughput screening workflows. Then Ian Russell, our Senior Product Manager at Labcyte, will explain the benefits of acoustic dispensing for compound transfers. And finally, Gene Ananyev, our scientific speaker from the Carbone Cancer Center at the University of Wisconsin-Madison, will explain his research and how the Mantis and Echo have been integrated into the AlphaLISA workflow in his lab to promote efficient and reproducible high-throughput screening. At any time in the presentation, please feel free to enter your questions into the GoToWebinar interface, and we'll answer all that we can at the end of the web event. With that said, I'll introduce Paul Ventura, our Director of Sales at FORMULATRIX. Take it away, Paul.
Okay. Thanks, Tom. Afternoon, morning, everybody. There we go. So I'm going to go through about 10 minutes, cover about 11 slides, just an overview of who we are, the positive displacement technology that we work with, and give you an overview of not only the technology, but the applications we support and how we dovetail nicely with the Labcyte Echo for miniaturized protein-to-protein assays. So who is FORMULATRIX? This is a quick screenshot even right off of our website. By nature, we are a crystallography company. So when you boil down crystallography, it's basically taking a series of reagents of varying viscosities, concentrations, and blending them together to promote the conditions for crystal growth. So we've developed a series of liquid handlers for this application, in addition to a suite of imaging systems in order to do the analysis. So over the 14 years of our existence, we've spun out a new microdiaphragm technology that's been incorporated into a series of reagent dispensers that will be the focus of today's talk. In addition, not to derail the subject too much, but we are launching a new high-throughput digital PCR platform that will be launched the first part of next year, and there'll be future webinars to discuss that instrument in the coming weeks. There we go. So who is FORMULATRIX? We've been around since 2002. We are a completely private-owned and employee-owned company. We now just broke 350 employees, so we're growing each year. We have an install base of greater than 1,500 instruments. And what I try to do in highlighting this slide is to give people the impression that we do have a global support network. So by having 1,500 instruments out there, imaging stations, liquid handlers, we have 3 different time zones of support and support coverage. So we are a world service organization. So the focus of today's talk is going to be 2 products that we currently offer in the reagent dispensing world. The Mantis, which is on the top right. The difference between these 2 platforms, being the Mantis and the Tempest, is purely throughput. They utilize the exact same technology, which I'll highlight in just a moment. The Mantis is more designed for low to medium throughput, people doing a lot of assay development work, well-to-well dispensing. And the Tempest is really the big brother to the Mantis in that it can offer much higher throughput capacity, where we can fill a 384 well plate in under 10 seconds. So it gives you a lot of speed, a lot of flexibility, but they both work off the exact same technology platform. Very low dead volume in our instruments, which has made it really amenable to an AlphaLISA type assay where reagents are extremely pricey. So Mantis can utilize a 6 microliter dead volume, whereas a Tempest can be as little as 40 microliters. As Tom mentioned when the talk kicked off, it's a positive displacement technology, and one of our biggest assets there is it's completely liquid class independent. So if a customer buys one of our instruments, they can use the same settings for a glycerol-based sample versus an ethanol or water. There isn't a lot of liquid classes that are very common with high-pressure Lee valve type dispensers or peristaltic, where the density of a fluid really determines its accuracy. All of those variables tend to go away when you get into our technology. It's also very little shear stress. And we highlight that because one of our bigger applications is cell dispensing. So we're comparable to that of a hand pipette. with 98% cell viability after a dispense through one of our microdiaphragm chips. So how does it work? So I'm going to pause, this is a video, but I'm going to pause it as it goes through to kind of highlight some of the features here. So this is a cross-section of one of our microdiaphragm pumps. Everything works on vacuum and pressure. So the first thing we do in our chips is we put them under vacuum, and what happens at that point is the inlet valve gets pulled open, so you'll see that. Here is our microdiaphragm. I'm just going to pause it. So what happens now is under vacuum, the diaphragm opens up, and this is a fixed container, meaning that it only holds a precise volume. So therefore, we get around liquid classes. So whether the sample is glycerol, whether the sample is ethanol or water, this chamber here that's being filled can only hold a very precise amount. Once that chamber is full, we then turn off the vacuum. We put on positive pressure, and that closes the diaphragm at a rapid rate, and therefore it will, you'll see in this video, eject a droplet out the bottom. I'm going to pause it for another moment, just kind of steer your attention to here, which will show you how this works in more of a live sense. So it almost acts like it's a breathing. So reagent's in, diaphragm closes. Reagent gets sucked in, the diaphragm closes, and that creates these droplets, which are extremely precise. Let's get to the next one. So I'm going to give you another quick video, because who doesn't like videos? And this will give you an idea of basically the dispense mechanism. So hopefully you're catching it in a live view, but we basically accumulate the drops until we get to your desired volume. Pause that for a moment. We also have the ability to dispense any volume into any well. So this is set up on a simple piece of plastic, but the idea is to show you that it doesn't matter what volume is being dispensed, doesn't matter what plate density you're working with, whether it's 24 well straight up to 1536, and it doesn't matter what volume you want to dispense into those particular wells. So a good example would be perhaps you're doing some normalization for a sequencing assay. Well, we can dispense any volume of diluent to any well, even import an Excel file to give it instructions on where to dispense your particular reagent. When you're done with that reagent, the chip gets dropped off, or in this in this example gets picked up. You can see it just broadcasting across a simple 384 well microplate. But again, it's precise drops that are overlaid on top of one. Very similar to the way the echo performs. You're looking at 2.5 nanoliter, 25 nanoliter droplet increments. We do 100 nanoliter, 500 nanoliter, 1 and 5 microliter increments to get to your desired volume. We also work with deep well blocks. So if you want to do something that required hundreds of microliters, that's an easy thing to put onto our platform. The standard Mantis can hold up to 6 reagents, or you can get a carousel that enables you to do all the way up to 24 and even 48 for certain applications. In this video, it's a really quick, like a simple setup. So what I wanted to bring to your attention is the ability for us to use a pipette tip as a reservoir. So let's say you have an extremely precious reagent, somebody gives you 100 microliters and you want to dispense 1 microliter across a 96 well plate. Well, it doesn't make sense to put it in anything that contains a lot of tubing, as you can see on the left-hand side, because there could be a more associated dead volume loss with that. So you can take a standard P200, P1000, simply aspirate up your sample or your actually your reagent, put it in one of our dispense chips, and then let the instrument do the rest. And that minimizes any of the loss that you would have. It also allows you to recover any unused material by simply removing the pipette tip and dispensing it back into your source container. So it allows you to really save on precious reagents. So as the arms, they just cycle through. What I always tell people is the Mantis just looks really good moving these chips around. And that's really what it is. The microdiaphragm pumps themselves is really the heart of the technology. Once it's done with the cycle, it just drops the chips off, and then it waits for the user to either add another plate for our next round of dispensing, or simply take that plate to the next step in your workflow. So what do these microdiaphragm chips look like? So this is a nice overview. So what I want to draw your attention to are these cylinders here. So these are our diaphragms. So this is a low volume chip. So this little circle here will hold about 100 nanoliters. This one will hold 500 nanoliters. So if you think of it in this context, this chip gets put under vacuum, 100 nanoliters fills in here, 500 nanoliters fills in here. We then shut the vacuum off, put on positive pressure, and these cavities evacuate, creating a very precise drop. and we replicate that drop to get to your desired volume. So if you wanted, let's say, 300 nanoliters, we would do 3 100 nanoliter drops all on top of one another. We also offer a high volume chip, which is the exact same technology, but instead of 100 nanoliters and 500, we offer 1 and 5 microliter drop increments. If you want to do really large bulk reagent dispensing, and when we mention large, we're talking hundreds if not mils of reagent. In the previous slide, we showed you a video where we can dispense into deep well blocks. So if your application required, let's say, 400 microliters, you don't want to sit there and have it do 5 repeatedly over and over. So we have a continuous flow, which dispenses at rates up to 200 microliters per second. And that's good for your cell medias, your less expensive or less precious reagents. That allows us to do very quick sample additions. To give you a sense of speed, 100 nanoliters can be done in about 25 seconds. That's the Mantis. The Tempest can operate that under 3 seconds. So you can tell that it's the Tempest is really designed for high throughput applications. These chips always comes up, are they reusable? They're about $60 US per chip. They are completely washable, completely reusable. We've tested them up to 2 million dispenses. And as long as, like anything else, as long as they're taken care of, they should last well over a year, if not the life of your instrument. CV data down below. Since we are doing a very precise droplet, our CVs tend to be average between 1 and 2%. Our specs are less than 3% for 100 nanoliters and 1 microliter dispensing. We also offer a variety of other chips. Let's say you're doing DMSO, we're doing a variety of organic solvents. We have organic resistant chips, and we also sell ones that are DNase and RNase free if you're in a pre or post PCR lab. Okay, next slide. Okay, a little lag. So what does the user interface look like? So when we sell a Mantis, I went too far ahead. Hopefully it'll back up. There we go. So when we sell a Mantis, we don't do installations. Because we have no liquid classes, it's a plug-and-play instrument. The operator buys the instrument, shows up in a box, they plug it in the wall, and plug it into a computer, and they're ready to go. So we designed it to make it a very easy experience for any user that's out there, whether they're an expert automation engineer or a very novice. They've only worked with handheld pipettes, and they want to take that first step into automation. This is a starter automation kit, if you will. So the software is all designed in a single screen. Basically, the first side of the screen, you're going to tell the Mantis where your reagent's located. So here you just simply enter in the type of reagent you're using, what place on the instrument they're indicated in. Here you'll enter the order in which you want your reagents dispensed, and ultimately how much volume do you want dispensed across a plate. These can be imported via an Excel sheet, or the end user can come up and simply click rows, columns, whole plates. So you do have a unique advantage in that every well can either receive the same reagent, or you can have a different reagent, or excuse me, a different volume in any well. But the goal is to have it all be on one screen and a very simple interface. So where do we sell, or where are our customers based? So wherever there's expensive reagents, we do a lot of business in the genomics space where PCR master mix, NGS library kits are very costly. We do well with cell assays just because it's a low stress dispense. But one of our other bigger applications is assay development. The ability to take between 6, 12, even 24 different reagents and combine them in any volume across any density well lends itself to an exceptional tool to really find the ideal conditions for whatever you're working with. Whether it's a signal and noise background you're trying to identify, this rather than doing this by hand, the Mantis or the Tempest can speed that workflow up intensely. So where do we, in a workflow orientation, so we partner with companies like Labcyte because we don't do sample transfers. So the Echo is first in class for doing low nanoliter transfer samples, but that's something that we can't touch. So when we sell our diaphragms and our technology, it's strictly for reagents because you don't want to have to go through and wash your chips before each sample. It's not very cost effective and it's not the right solution. So we do partner with Labcyte as an excellent choice for both nanoliter transfers coupled with our technology for all the reagent broadcasting on top of it. Again, there's no liquid classes, so depending on if you're doing bead addition or ethanol, it's not a problem, and you just tell the instrument what reagent you want in any well, and the instrument takes care of the rest. Okay. So who is our customer? So I mentioned just a few moments ago that we sell to labs that have a single robot in their lab. That's the person that's got a handheld pipetter that wants to get into automation or wants to miniaturize an assay but is not sure how to do that. So we can take the cost quickly out of somebody's assay by just reducing the reagents alone. They're still required with a handheld pipetter to do their sample addition, but it's a nice entry point for them to get comfortable into a automated platform. I can't tell me labs I've been into where there's an older liquid handler in the background that's covered in dust that someone had bought for a specific project and then either lost the expertise on how to operate it, or their workflow had changed and they didn't have the knowledge on how to adopt the technology for that. That's an ideal customer for us as well. So the Mantis is literally plug and play. Any user can learn how to use it in a matter of minutes. When it's not being used, it can be put in a closet, can be put in the back of the shelf, and completely be out of your way. The other side of our customer base are those that have fully integrated systems. So not every application or every day are you required to fire up your Hamilton system, for example, and put 20 plates down for a large screen. That's some days you just want 1 or 2 plates done, and our Mantis and Tempest are exceptional platforms for that. So our customer base really varies between the real tech-savvy automation engineers in a high-throughput lab, all the way down to your basic first-time user that just really wants to take advantage of a miniaturized assay cost savings, and that's pretty much our ideal customers.
All right. Well, thanks so much, Paul. That was a really nice overview of our liquid handling technology and how it benefits researchers performing ELISA assays. Next, we have Ian Russell from Labcyte. Ian has over 15 years of experience in the life science industry, and Ian joined Labcyte in May of this year and currently manages the Echo liquid handling product line, a series of instruments that enable the touchless transfer of nanoliter liquid volumes with extreme accuracy and precision. Prior to joining Labcyte, Ian managed a broad variety of novel technologies, including microarray and qPCR-related instruments and associated consumables. Ian completed his PhD graduate work with a focus on gene expression at the European Molecular Biology Laboratory in Heidelberg, Germany, prior to completing a postdoc at the Massachusetts Institute of Technology in the cell cycle biology field. So the floor is yours, Ian.
Thanks very much, Tom, for that very kind introduction. So I'm going to spend about 10 minutes giving you an introduction to Labcyte and the acoustic droplet ejection technology. And then I'm going to transition into sharing some of the applications that our drug discovery customers typically benefit from when using an acoustic liquid handling instrument. If I can get the slides to move forward, there we go. So in terms of Labcyte as a company, it was founded way back in 2000. So Labcyte has been around for quite a number of years. The first Echo liquid handler was placed in 2004, and that initial focus was on DMSO classes of fluidics, and really the early adopters were that early stage pharmaceutical discovery teams. Through working with those groups, the Echo, they had asked to improve the speed of the Echo transfer, and I'll get into a little bit of that in some of the later slides, but about 2 years later, Labcyte launched the 555 instrument, and that is a faster version of the 550. Then several years went by, and 2011 we had really 2 big changes or releases there. One was the release of dynamic fluid analysis, which enabled the Echo to address a number of different fluid classes outside of DMSO, and I'll go into some detail as to what that is as we get to the technology slides. We also launched the Access Laboratory Workstation, and that has enabled Labcyte to integrate the Echo into a larger workflow whereby the Access provides automation and the integration of various devices such as plate readers or plate sealers. With the improvement of dynamic fluid analysis and the ability to start addressing aqueous solutions, we started seeing adoption of the echo in the genomics field. And those customers typically were transferring larger volumes of fluids. And so in 2013, there was the Echo 525 that was launched that had a larger drop volume and just enables those customers to set up those larger volume reactions in less time. And then some of the current development projects that we have going, 2014, we initiated an Echo mass spec project, and that is still going. And we will be having some webinars on that, so look out for that. And then in 2015, we initiated a collaboration with Brooks to develop an acoustic tube and an echo instrument specifically designed to support that tube. And that's something that we anticipate launching early next year, and we'll be running again some webinars as we approach those dates. So please do look out for that coming. Okay, so I talked a little bit about the Echo itself, but that is part of the larger system solution that we have. The Access Workstation, as I mentioned, enables the Echo to be coupled with a variety of other devices and fully automated. And then we have a full suite of consumables that encompasses everything from a 6-well reservoir plate for bulk dispensing up through 384 and 1536 plates. And those come in a variety of different flavors, both standard as well as having low dead volumes, and can be purchased with a variety of different coatings on there depending on the applications that you're running. And then lastly, we have the Echo software applications, and I'm showing you in this slide a few of those there, and those are really aligned with the various applications that our customers are using, are very user-friendly, and use a wizard-based approach to setting up a run on the Echo instrument. The Echo liquid handler comes in 2 different flavors. We have the Echo 525, which transfers an increment of 25 nanoliters. It has the fastest flow rate, so that's 5 to 6 microliters per second. And really that's ideal for those larger reaction sizes, anywhere between 200 nanoliters up to about 10 microliters. And then the Echo 555 or 550, both transfer a 2.5 nanoliter increment. The 555 transfers at between 500 to 600 nanoliters per second. The 550 is a little bit slower than that. But both of these instruments are ideal for reaction sizes from 5 nanoliters up to about a microliter. The 525 supports a 384 well source plate format, so that would be the plate that your samples are being held in. And the 55X series supports both 384 as well as 1536. Both of these instruments support a variety of different destination plates that really only just have to be in an SBS format. So we have customers using PCR plates. We have a variety of different 1536, 384, 96. Really, if it's in an SBS format, then you can use that as a destination on the Echo. Let me talk a little bit about the technology here. So the technology is based upon sound energy or acoustic energy. At the heart of the instrument, we have an acoustic transducer. That transducer has a lens that enables us to focus that acoustic energy at the surface of the fluid. And through that focusing of acoustic energy, we can very accurately and very precisely eject a predetermined volume of fluid from that surface. So that's either going to be a 2.5 nanoliter droplet or a 25 nanoliter droplet. For transfers of larger volumes beyond that, Just like as you saw in the Mantis and the Tempest, we are actually ejecting multiple droplets to make up that larger volume. This is a non-contact dispense. There are no tips involved here, and I'll talk a little bit about what the benefits of that are in some of the later slides. But because that destination plate, which is up here, is inverted over the top of our source plate, so this is where the sample is coming from, it's being ejected into the destination plate up above, we can position that destination plate any well over any well of the source plate, and then the transducer can move underneath the source plate under any well. So we basically support any well to any well transfer, which is great for situations such as design of experiment or in some of the synthetic biology fields where customers are interested in dispensing multiple liquids into a single well. Let me touch upon dynamic fluid analysis, because this really differentiates the echo within the acoustic transfer field. So there's a video that's running here that demonstrates the energy that is coming from the transducer. We look at a variety of different echoes from that. So the sound energy bounces off the bottom of the plate. It bounces off the top of the bottom of the plate, and then you're going to see it bounces back off of the surface of the fluid. So just as the echo can be used to eject liquid, it can also be used, similar to sonar in a submarine, to start taking measurements of the fluid that is contained within that particular well. And through that, we can get a variety of different information that describes that fluid, such things as the fluid height. So it enables us to very accurately focus that acoustic energy at the meniscus here, as well as other characteristics such as DMSO, concentration, etc. And what that enables the customer to do is simply pick the gross fluid class that they have, so whether it contains glycerol, whether it contains surfactant, or whether it is aqueous or DMSO. And then the instrument itself makes all the fine tunes beyond that to accommodate different DMSO concentrations. If you have hydration going on during that transfer, it will adjust for that. And so there's very little calibration work that needs to be done on the part of the customer when using an echo to transfer their liquid. The reason that our customers are purchasing an echo is described here. It really falls into four main buckets. The first one is to maximize that lab budget through assay miniaturization. So the echo is very good at getting down to that nanoliter volume dispensing. It enables customers to conserve both samples and reagent. Because it's a non-contact dispense, there are no hit costs involved. And in many cases, it enables our customers to eliminate specific workflow steps from their experimental design. It also increases data quality, which results in fewer reruns. It eliminates pipetting variances due to fluids adhering to the inside of pipettes or carryover there. There's no sample loss on tips, no cross-contamination. And another big benefit here is we don't open ourselves up to any bioactive leachates that may be coming from the tips that we're unaware of. It's highly rapid in terms of that transfer, and it's very easy to design, test, and optimize the assays on the instrument. And then lastly, the ability to automate. So it enables transfer of multiple reagent combinations very quickly. And as I mentioned, if you are an assay developer, it enables you to do that transfer from any well to any well extremely rapidly. So let me change gears just slightly and share with you some of the applications here. This is from a publication that I encourage you to go out and take a look at. First author is Jacqueline Naylor and comes out of MedImmune. But essentially what the group here is doing is looking at a peptide-mediated activation, and both on a tip-based system shown here in panel A and on the echo in panel B. And what they experienced was dependent on which platform they were performing this on, they were seeing different EC50 curves and had hypothesized that this was due to the peptide adhering to the tips themselves. So you're looking at a panel here where they look at a variety of different BSA concentrations as a carrier here. And as you can see, as the BSA gets higher in concentration, so moving towards the DMSO here, it is helping to prevent that peptide from adhering to the tip, but it never quite gets to where the DMSO solution is itself. If we look at the same set of samples transferred on the echo, we get the exact same answer, irrespective of whether it's being transferred in an aqueous solution, a DMS solution, or a variety of different BSA concentrations that are there. So that's really huge in terms of ensuring that you are getting the correct answer from your dose response curves. One big advantage in the drug discovery or compound management world is the ability to do what we call direct dilution. Here on the left, we are starting with a sample in the square box, and this is the more traditional approach where you take a portion of that sample, you dilute it, and then from that dilution you take a portion, you dilute it again, and you're doing that serially all the way down. As you do this, we of course have many touches with tips, and any errors in dilution is going to be propagated across all of those dilutions. What one can do with the echo, because of its ability to transfer very small volumes, is do what we call direct dilution. So we take a larger volume from here, we go into the first dilution, a slightly smaller volume, go into the second, until we get right down to the resolution of the instrument at 2.5 nanoliters is your smallest dilution. What that enables one to do is, here we're looking at a standard dose response curve, an IC50 here. And this is an 11-point dose response curve. And the way that this has been set up on the echo is in creation of two intermediary plates. So we start with a 10 millimolar stock. We're making one intermediary dilution at 0.1 millimolar and then a third one at 0.001 millimolar. And then from each of those within the colors, here we're dispensing 25 nanoliters, 7.5 nanoliters, and 2.5 nanoliters to get our upper part of our curve. In the middle, we're dispensing from our first intermediary concentration from 92.5 nanoliters down to 2.5 nanoliters. And then for our lowest concentration points on the curve, we're dispensing from our most dilute intermediate from 150 nanoliters down to 5 nanoliters. And so you can see that we can do 25 microliter assay volumes here across a very broad dilution range and utilize only about 288 nanoliters of our compound. Before I finish up, I just want to touch upon the ability to miniaturize. This gives you a perspective on what our customers have been able to achieve in a variety of different areas, from PCR, qPCR, an HTRF assay, an ADP glow assay, the level of miniaturization. And this largely factors directly into the cost savings that they're seeing. So many of these are more in the genomic space, but we see anywhere between a 4x reduction in cost to 100x reduction in cost, and certainly within the drug discovery space and some of the more typical assays you might be doing, you know, here's a 4-fold reduction, here's a 10-fold reduction with those types of assays. So just to finish up, let me just summarize the advantages of the echoes. They're ready to go out of the box, so very little needs to be done in terms of getting it up and running. They are easily integrated into a larger automated system. We have a system from Labcyte that we can wrap it into, but we do also have customers that work with external integrators to do that. It is a contact-free transfer. It does not use tips to do that transfer. It enables the researchers to transfer from any well in the source plate to any well in the destination plate. Key areas that really benefits is in rapid cherry picking. And then I shared with you the ability to transfer without pre-dilution and some of the benefits in terms of assay miniaturization. So I'll stop there. Thank you for your attention, and I'll turn it back over to the organizers.
Hey, thanks so much, Ian. That's a really nice overview of the ECHO technology and the benefits. Next, I'm thrilled to introduce Dr. Gene Ananyev. Gene graduated from the Worcester Polytechnic Institute with a degree in biology. He received his PhD in cellular and molecular biology from the University of Wisconsin-Madison, working with Professor David Schwartz on developing high-throughput methods in genomics. As a postdoc, he worked on assay development for drug discovery using iPS cells, cell-derived neurons, with Professor Chang. In 2012, Gene took a position as a research scientist and manager for high-throughput screening at the University of Wisconsin-Madison Small Molecule Screening Facility. In the past 5 years, the focus of the facility has been the development of cell-based and biochemical assays in 1536 well format. So take it away, Gene.
Hi, how are you? So thank you for allowing me the opportunity to present. And today we're going to talk about some of the miniaturization of protein-protein interaction, high-throughput screening in 1536 well plates that we've done here at Small Molecule Screening Facility at UW. So first up, I'd like to introduce our facility and some of the folks that work here. So Professor Michael Hoffman is our faculty director. Myself, Song, and Ken work on the high-throughput screening aspect of the lab, and then Scott, Spencer, and Ken Sadashir work on medicinal chemistry and computational chemistry here at the facility. And we've been around for quite a bit. So we were established in 2003. We're a fee-for-service lab, and if you want to learn more about our services, this is our website. So the first question that we usually get when we do these kind of presentations on campus or off campus is essentially, why small molecule screening facility? What are small molecules? So a small molecule is defined as a low molecular weight organic compound that may perturb a biological process. So usually these compounds are under 900 Daltons. And why they're important is that most drugs that we have in use today are small molecules. So you can think about the small molecule as an exogenous ligand that binds a target protein and somehow affects its function. I do usually like to give a little bit of the background on the methodology of high-throughput screening, how the field came about, and some of its history. So the origin of high-throughput screening is really in the mid-1980s, and the origins are in natural product screening for antibiotics. Originally, labs were doing disk diffusion assays or liquid broth assays to inhibit growth of specific microorganisms, and the throughput was quite low. So you're talking about one extract is usually one test, on the order of milligrams of the compounds were used for these experiments, and even the best labs were only doing 20 to 50 compounds per week. So by mid to late 1990s is when you start seeing a lot of automation take place on this. At this point, compounds got standardized as being stored in DMSO, multi-well plates came on the scenes, and labs were doing about 10,000 compounds per lab per week. And the primary interest at that point was oncology targets. And then around 2000 to today, there's been a resurgence of going back to natural products and screening for antibiotics. But really, it can be defined by further miniaturization. So we're going to 384 well plates, 1536 plates, and now even lower formats. So at the heart of our facility, besides all the laboratory automation instrument, is our small molecule collection, which is can be called a compound collection. So we have about 500,000 compounds, all of which are stored in either 384 or 1536 well plates. So here you can see an actual compound plate with the different compounds in it. The majority of our compounds are what's known as diversity compounds, with the largest single collection that we have is the NIH's MLPCN library, which is 330,000 compounds. In addition to that, we have a number of smaller libraries, so all known FDA drugs, kinase inhibitors, anti-infectives, some natural products, etc. So really, for us as an academic facility, the question goes is we have this wonderful collection of compounds. How do we screen them for disease-relevant targets and do drug discovery in an academic setting with academic budgets? So I'm going to talk to you about a couple examples of protein-protein high-throughput screens that we've done for the laboratory of James L. Keck, who's faculty in biomolecular chemistry here at University of Wisconsin-Madison. And first, we're going to walk through a paper that came out in 2016 with the work that was done at the facility. And this is kind of one approach to doing high-throughput screening. So this was a fluorescence polarization screen for a protein-protein interaction, which is a Fanconi anemia. So this was done, well, I'll show you the screen setup a little bit later. So this is the actual setup that we had for the screen. We screened about 75,000 compounds in 384 well format using tip-based liquid handling. So in total, we used about 2.4 liters of the protein solution for the FT assays. And this screen was fairly expensive because of the amount of tips that we used and also fairly large dead volumes. So 5 to 10 mil per day of the protein solution was going to waste just on dead volumes. And here you can see this is the setup for 25,000 compounds. So these are all the tips that were being used, and actually the tips don't go into a landfill. They end up getting burned and incinerated because they're biohazard waste in the apex. So after the experience of this HTS and the data that we got, there was really a lot of interest from the Keck group to screen more compounds, but to do it with a more manageable budget. So this is a paper that came out of their lab in 2017 on a high-throughput screening strategy to identify inhibitors of SSB protein-protein interactions in an academic screening facility. And so the goal that we set out to do was to minimize cost in protein use and to come up with a cost-effective means of screening 100,000+ compounds. So just to give you a little bit of background, is one of the secondary screens that we used for the initial FT screen was using alpha screen technology from Perkel and Elmer. So this is a bead-based ELISA replacement where essentially you're doing a sandwich-based assay to capture an analyte using two beads, which when one of the beads is excited, it emits oxygen and produces visible light. So this is a very rapid assay. It's not labor-intensive at all, can be done in about an hour, and it's scalable to low volumes in 1536 well plates. So why 1536 well plates? Obviously, the answer is, because of size, but really on our end, it's because of volume. So here we have a two-scale drawing of the various well plate formats, so 48, 96, 384, and 1536. And volume scales accordingly. So by using 1536 well plates, we can conserve protein, reagents, miniaturize, and the cost savings are actually very significant. They're an order of magnitude, especially on the detection reagents such as alpha lysa beads or luciferase, anything like that. So for a 384 well plate, we're using anywhere from 20 to 60. For 1536 well plates, we're using somewhere between 3 and 5. Now, for anybody that's worked with 1536 well plates, doing this with tips is actually extremely expensive and cost prohibitive because you generally need proprietary tips. So at that point, our lab was fortunate enough to, through various sources of financing departments and grants and the facility itself, acquire an Echo 550 that we bought, and then we also have a Mantis instrument and a BMG Ferrostar plate reader. So the setup for the screen was to use nickel chelated and streptavidine donor beads for the alpha lysa assay and to miniaturize it using a synergy of these three instruments. So we used an Echo 550 for compound additions into 1536 well plates. So we're adding 10 nanoliters. For the master mix, which is the protein and alpha lysa beads, we were using 3 microliters per well. And then the detection was done on a BMG Ferrostar. And an important note here is because of kind of the Mantis's ability to use very low dead volumes, this helped us conserve reagents incredibly. And also we completely eliminated tips, obviously, using this methodology. So what is the kind of end result of it is that the protein use was scaled down by about an order of magnitude, so to screen a roughly equal size library. Compared to FT, we only used 240 mil of the protein solution. And for the bead use, So while the manufacturer recommends for 25 microliter reactions, we're getting about 10,000 data points per 5 mgs of beads. With the way that we were able to scale it down in 1536, we're doing close to 300,000 assay points for the same reagent. And most importantly, if you've ever made protein, that's a very labor-intensive process, is that the complete use of protein for this screen was only 5 mgs compared to what was estimated to be 1 gram for the FP screen. And since this paper came out, we actually now screened in excess of 500,000 data points for this target, and we're screening multiple other targets with this in other labs. As far as data quality and kind of what we came up with, so we screened 73,000 compounds with 9 hits coming out that are potent and selective. Z prime scores were all above 0.5, so the data quality is actually very, very good in this methodology. And so to kind of further continue on this topic, using a combination of the ECHO and the Mantis instruments, we've done alpha screen for several other targets. We've also done a number of cell-based reporter gene assays using nanoglow as a luciferase detection reagent and some biochemical screen for metabolic enzymes. And actually here is a picture of a recent paper, lab of Xinyu Zhao, also here at UW-Madison, reporting some 1536 well screens that we've done using iPS cell-derived neuron progenitor cells, actually. And so with this, I'd like to give you my contact info in case you have questions about assays or targets or anything like that, and just give you kind of a broader overview that, so small molecule is just one part of this larger drug development core here at UW. And all 3 of our facilities, so the small molecule screening facility, the cancer pharmacology lab, and the medicinal chemistry lab, offer our services to more or less anybody in the university system and outside of it for drug development needs. Thank you.
All right, thanks so much, Dean. That was a really great presentation, and thanks again for taking the time out of your busy schedule in the lab to be with us and to work with Labcyte and FORMULATRIX on this presentation. It's really nice to see your lab benefiting from this kind of novel approach and technology. So now we'll move on to question and answer, and we'll take any questions that have come in. So the first question I have here is for Ian. Ian, is the ECHO 21 CFR 11 compliant?
Yeah, thanks, Tom. So at this point in time, the ECHO is not 21 CFR Part 11 compliant, although we have a project that is nearing completion that will provide a new software application that will make it 21 CFR compliant. So I do anticipate that we'll, starting in November, start having some webinars that will provide additional information there. So do look out for that, and I believe that we're on track for releasing that software in the new year.
Great. And then for Paul, is the Mantis DMSO compatible?
Yes, we have a new organic chip that's out, so not only can you dispense DMSO, but you could also dispense acetonitrile and a variety of other organic solvents. That's the same performance that our normal polystyrene chips can handle.
Awesome. I have a question here for Ian. At which volume will drops fall down of the inverted well of the top plate?
Yeah, that's a great question that I think most new users to the technology think about. Because the destination plate is inverted over the source plate, obviously there will be a point in which the liquid will drip out, and that's very dependent on the type of plate, the dimensions of the well in that plate, as well as the fluid that one is transferring. I will say certainly for 1536 and 384, that's typically not an issue because those wells are quite small. It's mostly the surface tension that is holding the liquid in there, but we do also have what we call a high voltage grid on the instrument that helps the droplets to not bounce out of the well itself when they are transferred over there. When using a 96 well plate, particularly if it's a round bottom PCR plate, it is worth just testing that the volume of your reaction that you're setting up, you won't have that issue with. I will say that we have many, many customers who are using the Echo to dispense into 96 well plates for setting up qPCR types of experiments, and for the most part, that is not an issue at all.
Great. Thank you very much, Ian. I have a question here for Gene. Did you notice a difference in activity between a dry and wet dispense of compounds? Dry dispense compounds and master mix on top versus wet dispense of compounds on top of master mix?
Not really. So we've done some testing for that, and in our hands does not make a difference actually for most assays.
Great, thank you, Gene. I have a question here for Ian. Is the Echo able to dispense cells from quite a concentrated suspension into a 1536 plate at about 1 cell per well concentration?
That's an interesting question, and the short answer is yes, it is able to transfer cells. And we certainly have customers who've used it specifically for that application. It does depend on getting those cells into suspension and maintaining them in suspension. We actually have an application note that is up on our website using a variety of different approaches to how one might achieve that. So I'd encourage you to go to labcyte.com, take a look at some of the application notes that we have there that go into a lot more detail. But yes, it does enable you to transfer cells. And in fact, I think the other question that typically comes up in conjunction with that is whether the sound energy is gentle enough that it's not going to impact the cell itself. And the answer to that question is absolutely. Those cells that you transfer using the Echo remain viable. It is a very low energy acoustic burst that is emitted by the instrument. So it does not denature proteins, it does not impact DNA, and it does not impact the viability of cells.
Awesome. Thanks. I have another question for you here, Ian. Are you aware of any seeping issues of compounds added to a dry plate?
Any seeping issues? So I'm interpreting that question as in a similar fashion to the previous one in terms of the solution dropping out of the inverted destination plate. Again, no, there's very little issue with that. But it is going to be dependent on the fluid type and the dimensionality of the wells. So again, I would encourage a user to do a prior test just to ensure that that isn't going to be an issue. But it's probably worth mentioning at this point that we do have an extensive support group, a support team, very talented FAS, and they work directly with our customers to help get assays up and running. So if you have concerns like that, they have a depth of knowledge, and I would encourage any customers to reach out to them, and they can help you work through those types of concerns.
Okay. Thanks so much, Ian. I think that is that's all the questions we have time for today. So thanks again for all of our speakers for taking the time to share knowledge about the technologies that are out there for efficient and reproducible screening of compounds, and for Gene at the University of Wisconsin for sharing his profound experiences and cost savings in a kind of a consumable-free and miniaturized approach. This webinar will be made available as an on-demand recording and will be sent out to all attendees following the event. Thanks so much, everyone, for taking the time out of your day to be here with us, and we look forward to seeing you at future events.